one of the kind of strategic focuses that COVID has kind of brought attention to is that of repurposing, repositioning as a strategy, especially within NTDs. And look at the interplay between COVID, NTDs, repurposing, from a strategic perspective, but also from an operational perspective. So that's what the session is kind of all about this afternoon. I'm very, very honored to be joined today by the panelists. We've had the DNDi feature all the way through the proceedings this morning, from the reimagining financial incentives all the way through to the access and redefining access. So I think it's very apt and very right to have the world's leading PDP active in so many different diseases. It's such a huge success story to really lead the charge, as it were, in terms of repurposing as a strategy. And so we're very honored to be joined today by Dr. Nathalie Strub-Wourgaft, the Director for Neglected Tropical Diseases at the DNDi in Geneva. I'd like to pass the floor over to Nathalie in terms of her particular presentation, which is looking at COVID, NTDs and repurposing, the questions and lessons so far. So I think a round of applause for you, Nathalie. And if I could ask the other panelists videos off, that would be perfect. Thank you. Over to you, Nathalie.
Oh, thank you for the introduction. Let's see how it goes. It's I think we're very happy to be, to have the chance to sort of discuss. It's more discussion. What do we know so far? What are the questions that COVID poses on NTDs or NTDs on COVID? What have we learned so far? And then I'm sure we'll learn more, and we'll have to meet next year to see, take conclusions. So I just want to run through some ideas. It's nothing fixed. It's something that's in, the fact that COVID is everywhere, that we say that we're COVIDizing everything, and how we can connect back to NTDs. That's a bit the idea of this presentation. So maybe to start with, as you said kindly in your introduction, so DNDi genesis was in fact related to the lack of access to essential drugs for MSF doctors who couldn't find solutions to treat their patients. And they received the Peace Nobel Prize and then decided to engage in some work for neglected tropical diseases and find solutions. But what happened in 2020, and this was in 15 years back, but what happened in 2020 is that in a similar way, we looked at what was happening. Some of us researchers look at what was happening in low and middle income countries. Was research gaps going to be addressed for COVID? Was there attention? Was there not a risk of neglect for the needs of LMICs? And at that time, in March of last year, it was very clear that all the research was taking place elsewhere. So there was a kind of neglect area of research, and also a need for doing more for COVID. And obviously, this is how DNDi, with many others, we proposed a call for coalition of the willing of researchers to promote the needs and find ways to engage into research for COVID. So this is a bit the link. And I think the fil rouge, if you want, the red thread in this presentation is how we have parallels between NTDs and COVID. So what is the potential, what are the potential interrelations between COVID and NTDs and repurposing? So, the pillars of what we're doing in public health, we do R&D, we do this for public health impact, we do this with health systems, and we take care of patients. And we have in mind the economic impact of what we do and why we do this for those populations. And repurposing is about adapting what we were doing for NTDs or what others are doing for NTDs for use in a different purpose, in the very large sense of things. So if we start looking at care and health structure and stuff, how has repurposed, how has it taken place? And I'm sorry that we couldn't have WHO because they are really they've done a lot, and they are the first front runners in having to manage this. But as we all know, resources have been repurposed to manage health issues with COVID, and to look at ways of, for example, doing the social isolation and everything. But there's been a negative impact, clear negative impact also. As we know, where some of it's written here, some of the mass drug administration programs have been put on hold. Some of the active case management, case screening, have also been put on hold to manage the crisis. And so this has had a really negative impact on NTDs. Maybe a positive one is that there are more, hygiene, health, wash measures and all of that may have helped, but we'll have to measure the impact of this. Nevertheless, it's really impressive to see that, the WHO NTD set up different initiatives to manage and try to understand how to manage the impact of COVID on NTDs, and how could the NTDs and the COVID, the stuff, be repurposed, but also the future of not abandoning the NTDs priorities with the roadmap in mind. So that is one example and one impact, reprogramming, repurposing stuff from NTDs to COVID. Another question that we can ask ourselves is, do we know if there are any medical interactions of coexisting, having, for example, COVID and one NTD? And our colleague from Argentina, Sergio, with colleagues, published a paper looking at different areas of potential impact of COVID on Chagas. And they identified the fact that there might be some potential reactivation of chronic patients with chronic indeterminate Chagas that could be due to the virus or to immunosuppression, or even to some drugs that would be given to treat Chagas. They also envisage that there could be a direct impact of SARS-CoV-2 on the cardiac tissue, and all of that could impact Chagas. They also made some recommendation of how to treat different stages of Chagas in different patients. So that was one interesting publication looking really at the interplay between the two diseases. There's also a team in Nigeria that looked at the effect of schistosomiasis and treatment of schisto on COVID. And what they identified was that schisto could increase the rate of unfavorable COVID-19 outcomes, but conversely, that treatment for schisto could decrease the active COVID cases and increase recovery rates. So also another way of looking at the interplay between the two. Now, in terms of our work on clinical trials, and my colleagues have been before and will come after to speak about their own work. But yes, it has had a time effect. We have experienced delays in the work we are doing for NTDs. So just purely on R&D, we have had impact on achieving our ability to provide solutions due to COVID. We've had significant delays in phase 1 studies conducted in EU, in fact, I should say in the UK. We've had delays in recruitment of ongoing clinical trials in some of the regions, for example, in Sudan on mycetoma, due to either the program stopping or the quarantine measures or the isolation, so patients couldn't travel back to the sites. So they couldn't they couldn't be recruited. We've had delays in contract signatures because people were busy with other priorities. We've had some health staff that was infected by SARS-CoV-2, so that definitely impacts, and I mean in the hospitals. And in one of our study in sleeping sickness, the active case finding has been slowed down, so that has impacted this program. Now, we have described before the value of repurposing for NTDs. So do we, is is this still also of interest for COVID? And the value of repurposing is that you save time by by having drugs that have for which you have already a lot of information in terms of their safety, because you have already manufactured them, because they have been most of the time already registered. So a lot of things that take so much time during the value chain are done. And what you have to do is just go for a new indication. And so you will save tremendous time, which is critical in our response to COVID, but also you will reduce costs, which is important too. And so, yes, of course, everything has been around repurposed drugs, most of it. So here you have a nice publication looking at how machine learning has been, and don't ask me how it works because I don't know, but what you can see is here you have the list of the drugs, and you see here the different, what they call the hallmarks of what kind of efficacy they could see, and then they draft some radar, some presentations where they see the value of each of the drug. So that was one way of looking at repurposing. And this paper showed that, you know, which were the active drugs that have been repurposed for COVID. So a whole range of drugs from different categories. And here you can see Ivermectin as one of the candidates. So just one example, because this example of Ivermectin is quite important, because Ivermectin is, of course, a treatment for NTD that's repurposed for COVID. So where we stand now is that the story of Ivermectin, I'm sure you know, but it was discovered in the 70s in Japan by Kitasato Institute. It was first used for animal health, and then repurposed for human treatment of onchocerciasis, and then with TDR, and then later registered for other indications that are listed here. Now, the interesting thing is that it's been now largely tested for COVID. In fact, at different stages of the disease, either as prophylactic treatment, either for the treatment of mild or severe cases of COVID. You can see here that there's been it's a preprint of a meta-analysis already of Andrew Hill on the effect of Ivermectin on COVID. And I just checked this morning, there are still 37 studies looking at Ivermectin for various populations of patients with COVID. So clearly a huge impact of a repurposed drug that could become one of the tools to manage COVID. But there are some questions that we need to think about because of this repurposing effort. The first one, looking broadly on the public health consequences, well, it's very good if Ivermectin can be used in regions, for the world, but what will be the impact on the MDA programs? Should we wait? Do we have, in fact, enough capacity to manufacture, to manufacture Ivermectin at scale? And what will be the impact on the MDA program? And I have to say that with the response to COVID, there is the ACT-R, this big initiative regrouping the big donors and WHO to manage all of this and globally, that is really looking very in details of how this can be achieved, and they will find a way, I'm confident. Then the regulatory aspects, we shouldn't, forget about that. So it's probably going to go through emergency use listing, but then how do we manage the fact that most probably we will be using doses that were not the ones that are used for onchocerciasis? Are we going to learn something about the mechanism of action of Ivermectin? I was just thinking, well, maybe we'll learn something that will help to repurpose it for another indication, who knows? And of course, we also have to think about the impact on the co-infected individuals. So I'm almost finished. This is the slide that describes the partners that are working on NTCOV, which is a study where we're working with colleagues here on looking at a COVID response for outpatients. And what I couldn't do, because it would have made the slides even more busy, is in fact to highlight that each of these institutions that is working on COVID now was also and is also historically has been involved in NTDs. So even at the institutional level, DNDi, yes, but many others, all those for which you see the logo here, almost all of them, they were working on NTDs. They've repurposed themselves to COVID. And finally, something which is so worrying, that, you know, we know this vicious cycle of poverty related to NTDs, the poor patients being at risk of having an NTD, being then more impoverished by the NTD, which puts them back to even more poverty. And there was this report from the UN that also looked at the impact of COVID on poverty. So I think it's a double threat. It's a double threat to some of the populations, and we can't just separate them. They are even more vulnerable than ever. So this is something I just wanted to highlight, because it's so devastating to see that the impact is always on the same ones. So can we conclude anything? So I think so far the good news is that we can see that, you know, when you have a pandemic like this, you can mobilize resources. And in fact, you can make things happen very fast. We've seen incredible progress. The solidarity, the recovery results are just incredible. The speed of the vaccine, that's great. And that's quite encouraging. And I wish we could see the same, in fact, for NTDs. And in fact, the other thing is that we see that for NTDs, we've been very much looking at the same strategy of looking at repurposed, having short-term, medium-term, long-term approaches, which is exactly what's happening with COVID. But there are also some negatives, and you know, the impact on the healthcare workers, the fact that resources could shift, the fact that ODA funding has decreased, and that can impact our chances. So we have to be very vigilant and just keep communicating about not forgetting about NTDs in the context of COVID. Lastly, I've added 2 publications which I found really, very interesting, and no time to speak more. And I think I'll stop here. Thank you.
Thank you very, very much, Nathalie, for that. That was exactly the opening we were kind of hoping for, because it really set the scene in terms of the relationships between economic impact, R&D, healthcare systems, public health, the COVIDization of everything. And we really feel that it really actually speaks volumes to the effort behind repurposing, and it's actually going to be a very positive thing moving forward. And I think you really picked up on this kind of mobilization that's needed to do that. And building on what we've heard earlier in the morning sessions and today, just now, and leading into the next presentation, I think that this mobilization is going to require far better coordination between the different groups and the stakeholders involved. And I wonder what type of new tools are going to develop to bring that cohesion around the different groups together in this repurposing effort. And by that, I mean, you know, the kind of rapid kind of sharing of information, which potential therapies work, almost anthropological data at some level, but a lot of it really serving in terms of rapid communication, you know, case sharing, discussion. You put those 2 papers at the end, latest literature, and that kind of brings me in to the next. And I'd like to thank you, Nathalie, for opening up those avenues of discussion, which I'm sure we're going to revisit without any questions on machine learning. By the way, I won't do that to you, but I'm sure we'll come back to the Q&A at the end. But I think thank you very much for that. I think that's really opened up the floor to the next speaker. We're equally as honored and delighted to have on board the US Food and Drug Administration Cure ID project. Dr. Heather Stone from the Health Science Policy Unit there is going to be explaining what Cure ID is. We've heard the need for a cohesion between different groups. Look what's happened in terms of when you do get that cohesion with COVID and the development of a vaccine being cut from 10-year period to about a year. A lot can happen. And let's see if we can apply this in NTDs. To do that, we're going to need certain tool sets. And I think this is a fascinating development, Cure ID. I know my colleague Marianne Compre had a ISNTD Connect session earlier in the year with both Dr. Stone and Dr. Strub-Wourgaft and Nathalie there earlier. I think building on that and what we've just heard, I think I'd like to introduce the floor to Dr. Stone.
Thank you so much for having me. I'm delighted to be here. As said, I'm Heather Stone from the FDA. I'm an infectious disease epidemiologist and work in health policy. And delighted to be presenting today about the Cure ID platform, which is an FDA and NCATS NIH, the National Centers for Advancing Translational Sciences at the National Institutes of Health, collaboration, and then the role of the public-private partnership that we've developed with great support from Nathalie and others called the Cure Drug Repurposing Collaboratory. And building off of Nathalie's presentation, again with this theme of how have COVID and NTDs interacted with each other in the space of drug repurposing, both helping and perhaps hindering one another. So just a few disclaimers. I represent the FDA, but I will also be talking about the public-private partnership, and there will be significant discussion of unapproved uses or off-label uses of the drugs. So the first question is, what is drug repurposing? And you can define it in many different ways, but for the context of this conversation, I would define it as the identification of potential novel uses of existing drugs. It's our belief that for diseases lacking adequate approved treatments, clinical experience of how clinicians are using these drugs in the field, as it were, could be collected and used to identify promising candidates, drug combinations, treatment regimens, et cetera, in a much more rapid way that could then be used to inform clinical trials. This could include new indications or new ways of treating diseases, but also, as I said, new combinations, dosing regimens or durations of therapy, new populations, and importantly, unapproved uses that do not work or even harm patients. So this is not only identifying positive instances of drug repurposing, but also instances that have tried and failed. So in 2012 and 2013, FDA and NCATS got together and established Cure ID, which was a platform for collecting case reports from clinicians, as well as a discussion forum and mechanism for sharing journal articles, news articles, events, etc. The goal of this platform was to enhance the understanding of new uses of approved medical products, to facilitate clinical trials and drug development, and ultimately to serve as a resource for physicians to share information where no FDA-approved product, which had been proven to be safe and effective, already existed for the new use. Please feel free to visit Cure ID. It's available at cure.ncats.io on the web, or you can download the mobile app, Cure Space ID, in the Google Play Store or the Apple App Store. And it's freely available. So there are a number of ways that, or a number of specific areas that one can imagine repurposing having particular advantages in, and a platform of this nature, including neglected tropical or infectious diseases, but also antimicrobial resistant organisms, as well as emerging threats. And so we identified a list of initial priority diseases, including COVID, but also atypical or non-tuberculous mycobacteria, drug-resistant gonorrhea, mycetoma, rare and resistant fungal infections, and multi-drug resistant gram-negative bacteria. And then in the NTD space, more specifically, also questions about toxoplasmosis in pregnancy, leishmaniasis, Buruli ulcer, babesiosis, and dengue and viral hemorrhagic fevers. The platform works by having clinicians or other healthcare providers submit a very easy, rapid case report. It takes 3 to 10 minutes to enter via the app or website with completely de-identified data, so no patient identifiable information, and using largely standardized terminology. Once the case is posted, it's rapidly reviewed by clinician moderators, typically within 12 hours, often much less than that. And the moderators that have the option to contact the user for additional details or clarification, or in a very rare instance where the report is needs to be removed to do so. The data from the case reports is then aggregated to show the total number of cases using each drug alone or in the specific combinations of drugs that it may have used, and then can be further broken down by treatment outcome to try to help users have a sense of which drugs look the most promising or the least promising and how they compare. And then users can also download the entire de-identified data set to conduct their own research. We believe Cure can serve as a rapid communication platform for healthcare providers during an outbreak, providing for systematic case sharing, discussions, and sharing of the latest literature. It can facilitate sharing of information on the use of potential therapies for diseases which lack adequate approved treatments, and inform current and future clinical trials, serving as an important sort of bridging step between unstructured anecdotal reports and the robust randomized trials ultimately required for drug approval and strong evidence generation. Importantly, I think this enables the exchange of opinions from global communities of experts and helps break down some of the geographic silos that exist. And our hope is that it can help to identify potential new uses for existing drugs while limiting unhelpful or harmful uses. To date, we have included 325 different infectious diseases in the CureID platform, about 2,700 case reports, nearly 7,000 activated users, and more than 20,000 clinical trials. For COVID specifically, we have 1,100 case reports, which are a combination of those from that clinicians have submitted, as well as those in the published literature that our team has extracted. More than 100 discussion posts, 1,400 clinical trials, and 1,300 journal articles are amongst the information that's been shared. In total, 290 repurposed drugs have been identified in the CureID COVID case collection, and 45 of those drugs have 10 or more cases. And I'd like to specifically thank Dr. David Feigenbaum's Corona team at the University of Pennsylvania and the Castleman Disease Collaborative Network, which has been, as well as our fellows and interns who have gone to extraordinary lengths to identify these cases and enter them from the published literature. However, it's important to acknowledge that there are major limitations to this data, and in particular, the relatively low number of clinician-submitted reports and those in the published literature, when coupled with the high spontaneous recovery rate in COVID-19 and differences in severity of infection, it makes the data difficult, if not impossible, to interpret. So our solution was based on how can we get a much larger number of cases such that we would have more power to detect possible differences. And that seemed to require inclusion of a method that didn't require clinician entry. And so our proposal was to get that from electronic health records and other existing registries to sort of pull many different data streams together. Because the data we collect is relatively limited, it becomes possible to aggregate across many different streams of information. So we proposed an idea to build an automated extraction tool that would pull de-identified data directly from electronic health records and various registries into the CureID platform. We created a partnership between FDA, NCATS, and the Critical Path Institute, which houses our Cure Drug Repurposing Collaboratory, as well as 4 primary institutional partners, Johns Hopkins, Emory and University of California Health Systems, and the Society of Critical Care Medicine's Virus Registry. With this proposal, high-level case reports translated from EHR and registries to the CureID case report form at the institutional site with completely de-identified patient-level data would then be made openly accessible for use and download via the CureID platform. in a way that it currently is not even in many of the systems that do try to pull EHR data together. Sites that use a common data model would be funneled through the N3C collaboration at NCATS, where they have a mechanism for pulling that data together already. But importantly, sites that don't use a common data model, which is the vast majority, would use an edge tool developed by Hopkins that would sort of serve as a translation between a non-data model electronic health record system and the CureID case report form. And they would do that through the Society of Critical Care Medicine's virus registry. So the goal of this EHR data extraction would be to share data, making a limited high-value set of case data on drug treatments for COVID-19 openly available to all individuals. To work with Hopkins to develop this automated data extraction tool, which could then be shared openly and used for other purposes, and to provide this technology to further adoption by the SCCM virus sites to reduce the burden of case sharing. Those participating in that registry currently spend on the order of an hour and a half per case report entering the data. So you can only, and they have something like 65 or 1,000 cases at this point. So you can only imagine the burden of on healthcare workers to enter that information. And then ultimately, thinking ahead to facilitate the development of a sustainable infrastructure that would enable clinical care and clinical research to be more seamlessly integrated. So where we are now is that we have a working group of SCCM Mayo virus experts, as well as leading clinicians from Hopkins, Emory, and UCSF that have been adapting our CureID case report form for COVID for EHR registry data extraction, which enables us to get a lot more data than when we were asking for it from clinicians. Extending the partnership to include other health systems and registries, and then including an international component in partnership with the Infectious Disease Data Observatory at Oxford. This is just the structure of the Cure Drug Repurposing Collaboratory Coordinating Center, Partnership Coordinating Center. So you can see the main institutional partners, the PIPs as we call them, primary institutional partners, and then the large number of secondary institutional partners, all told being about 300 sites. So our hope is that within the next just probably 3 to 6 months, we will have included 100,000 patients de-identified data as case reports and made that openly available on the CureID platform in partnership with our partners. And within 2 years, which is a grant period that we've been proposing, we estimate to include 250,000 patients. And as I said, really looking to develop this as a sustainable infrastructure for capturing and sharing information, both through these registries as well as through this open source edge tool. This all is reliant upon a public-private partnership that enables us to work with stakeholders across a wide swath of organizations. And importantly, is really focused on how we move from anecdotal observational case collection, which is important but has limitations, to evidence generation and dissemination. So in June of 2020, the Critical Path Institute convened a public-private partnership in collaboration with FDA and NCATS and the CureID platform called the Cure Drug Repurposing Collaboratory. It has begun with a pilot focused on drug repurposing for COVID-19 and is also pursuing repurposing opportunities for several of the neglected tropical diseases and other infectious diseases in the previous slides. And our goal is to build infrastructure that could be used for COVID-19, but also could be expanded to other neglected infectious diseases in the future. And to really demonstrate how data shared from clinicians in real time can be used to inform future and ongoing clinical trials, and based on those trial results, potentially drug labeling or other regulatory decision-making. And this is an active group that is seeking partnership both from individuals and institutions. So I encourage you to reach out today, and there's contact information in a later slide. And these slides will be made available. Oops, wrong button. I won't go into depth, but just to show you the structure of the Cure Drug Repurposing Collaboratory, which includes an advisory committee that oversees a broad range of activities across drug repurposing in many different spaces, including infectious diseases, and now we're expanding into rare diseases, special populations, including pregnant women and neonates, and then some of the operational working groups, such as preclinical to clinical translation, clinical trial implementation and infrastructure building, and regulatory and legislative and policy. So just as a final thought, I think there is an opportunity to build infrastructure for COVID-19 that can be used for NTDs and other neglected diseases, and we need to take advantage of that opportunity. But I think COVID has also very clearly demonstrated to the world the need for collecting anecdotal and observational data, but for the purposes of generating hypotheses, not for the purposes of generating confirmatory evidence. And that observational data then needs to be rapidly and systematically translated into randomized clinical trials, and that these efforts need to be coordinated through groups like the Cure Drug Repurposing Collaboratory, as well as other public-private partnerships and institutions. So I encourage you to get involved in the Cure Drug Repurposing Collaboratory and or the Cure ID platform. Please feel free to reach out, and would like to thank our many partners for their support and participation, and our extensive Cure ID team and CDRC team, which luckily seems to be growing on a daily basis. So thank you so much.
Thank you very, very much for that, Heather. That was fantastic. I say that not lightly, because I say that from seeing all the ISNTD Connect sessions that we've had since March to now. This one really stood out for us simply because it is the future of sustainable infrastructure development when it comes to joining clinical care to clinical research. And that's all echoed in the comments we're getting. We're getting you asked for partnerships. Dr. Emmanuel Agunloye from the IBAN Food and Health Research Center in Nigeria. Can I share the Cure ID site to doctors in our community via my NGO site? I see you responded there. They're fantastic. Also, from the Ministry, from the National Institute of Health in Portugal, Dr. Maria João Gouveia was also saying this is such a fantastic tool, a very interesting tool. So I'm sure you'd have some inputs from there as well. You mentioned some of the diseases that you're active in terms of the layout and how Cure ID is being used. I definitely add snakebite to that, because you probably saw in the previous session, they're also actually opening up repurposing using HTS to see potential new avenues for small molecule inhibitors, and linking that to their strong clinical presence on the ground in the various African countries that they're involved in. And I think all of this adds to the flavor in terms of redefining access, giving that community a voice that's going to then ultimately impact the R&D processes. In a way, it's like digital pharmacognosy. And I'm not an ex-pharmacologist, and I kind of see it in that light. I think it's wonderful. It's brilliant. So I think round of applause. Jutta Reinhardt-Rupp from Merck saying thank you for this interesting presentation. I think it is absolutely fascinating, and it's exactly what's needed. We've heard people say transparency. Are we singing from the same hymn sheet, the right stakeholder mix? This is the key stone, we feel, for that particular part of the puzzle. So a massive round of applause. That brings me to the next speaker so thank you, Heather, for that. And that brings me to the next speaker for this particular session on repurposing. We've heard from Nathalie in terms of the large kind of engagements that COVID is having on public health, economic impact, R&D, the healthcare systems, and the interplays between them. We've heard from Heather in terms of the kind of an operational step, this Cure ID step that's needed to bring people together and channel that kind of information forward all the way to RCTs. randomized control trials, the gold standard. So we thought it'd be a good bridging, how to put it, presentation if we brought someone in to talk about the drug that's on everybody's mind. I don't think you can escape anywhere in NTDs for the last year without hearing the new role of ivermectin in a new disease horizon, which is fantastic. So I think it's the perfect time to bring in Dr. Carlos Chaccour, he's a Chief Scientific Officer at BOHEMIA project at ISGlobal in lovely Barcelona. He's going to talk about ivermectin and COVID, what's going on. Yeah, it sounds like a song, but what's going on is the title of that. So I know we've had some issues on sound, Carlos, so I've got fingers, everything crossed here for you. I think I'll just see. Yeah, brilliant. We can hear you. I'm going to just pass the floor over to Carlos and Dr. Chaccour. Thank you very much. And I'll ask everybody else if you could just mute yourselves, the speakers on this. That'd be great.
Great. Okay, I'll go ahead. So a lot is going on ivermectin. Thank you for the invitation. So basically, I'll try to talk around. I'll start with key messages. So we have the core concepts clear at the beginning. I'll follow with a landscape of what is happening, and then I'll try to provide some reasons as to why this is happening, what's the science behind it, and then finally my own views on what this is for NTDs. So the take-home messages, I have 2 slides. First, and everybody knows, several countries have already approved ivermectin or even rolled out ivermectin as a treatment or even prophylaxis for COVID-19. That means it's included in several national guidelines. However, the evidence base to support this is limited. There's also abundant misinformation, and some of the reasons behind this is initial contempt, or what I regard as contempt by some key opinion leaders, funders, international groups. Then a lot of social media pressure, advocacy groups, and there's even a role for fraudulent data on something that happened last year. The evidence that is coming up, there's both, there's positive and there's negative, but it's slowly emerging. And now WHO is looking at this under the guideline development group. The second part of this is that this is of course leading to a surge in demand, which is fueling profiteering. It's fueling the proliferation of low-quality products, fear of potential rerouting of donated product for the MEC examination program into this new, not yet proven indication, and concerns about sourcing API. And then there's also room for multiple doses or high doses being used, and there's concerns about maybe this will open or show previously non-seen adverse events that could affect acceptability of NTD programs. And then I have a slide at the end about optimal dosing and what's the interface between the diseases. So what's happening? Peru was the first country to include this in the national guideline for COVID in May 2020. It followed by seems to have canceled the program in October that same year, although it's still widely used in the country. Bolivia followed suit in May 2020 as well, and they even rolled it out to as much as 300,000 people last year in a single shot in one city. Brazil, being a federal state, has not a single national guideline, but it's been rolled out officially in several municipalities across the country. Venezuela was one of the latest in Latin America and included in the national guidelines on July 2020. And then this year, we have Slovakia, including this about 3 weeks ago in the national guidelines. On the same day, South Africa did it, and the next day, Zimbabwe followed suit. So a lot of countries rolling it out. The other component is in misinformation, and I'll just give 2 examples. One is very recent, by the way. This is from last week. An Australia medical group received a cease and desist order to stop promoting unproven therapies. And then this one's from Korea, in which the National Regulatory Agency closed down 750 illegal online websites selling drugs, including ivermectin, for the treatment of COVID. There's been a series of statements from authorities, both national, international, and even from Merck about this. The first one came out in April 2020. It was the US FDA saying, please do not use animal ivermectin for human use. PAHO came out in June 2020 saying there was not enough evidence back then. This may already be outdated. MEC design donation program with the statement 3 weeks ago saying, reminding partners please not to use donated product for other indications rather than oncocercic and LF.
Oh, no.
Then Merck came out just 2 weeks ago with this statement saying that there's no scientific basis for ivermectin and COVID, and there's a concern about lack of safety data. Finally, NIH had revised their clinical guidelines several times, and just 10 days ago, they has said there's not enough evidence to recommend for or against ivermectin and COVID. So why? Why do we have countries rolling it out, misinformation, and all these public statements? And I think there's a lot of reasons. I mean, there's a public health emergency of international concern, limited treatment options. We have policymakers and frontline healthcare workers desperate for options of treatment. And I'll try to put some other reasons on why this is happening, right? And the first is this in vitro data. In April 2020, there was this paper out from this Australian group showing that ivermectin can inhibit the replication of COVID-19 in vitro at 2.5 micromolar, which is way above physiological concentrations. And this led to what I regard as contempt. Many people came out to say the concentrations were too high. There's not even reasons to do a trial. When actually, when you have a disease that has no treatment on one hand and a drug that is quite safe on the other, generating evidence couldn't harm anyone. So I think there was an opportunity to let that speak, but that was not what happened, with many experts coming out saying that this is a shot in the dark. You should think twice before doing trials, and even mockery was out there. Another ingredient was the Sergio scandal, and for those that don't know it, I would recommend you read the timeline here, beautifully written by Catherine Offord in The Scientist. But basically, an apparently fraudulent database ended up causing simultaneous expressions of concerns in the Lancet and New England Journal of Medicine on June 2nd last year, followed by simultaneous retractions in both journals. But what less people know is that the same group had a preprint that came out in April last year, saying that ivermectin would reduce in 80% mortality of patients in mechanical ventilation. Another ingredient is the Frontline Critical Care Alliance, which is much more new. And this is a group of frontline healthcare workers that I think are generally worried about how to help people, and they have done their own evidence review. The bit that is more contentious is that they claim that there's no need for additional evidence. In fact, they claim that conducting trials would be unethical because evidence out there is already overwhelming. This was presented in the US Congress in December, and this hit the media quite strong. And since created this second wave that we saw, remember the countries I showed, there were countries in Latin America putting it out in May, June, July last year, and now countries in January this year. Interestingly, the Frontline Clinical Care Alliance also quotes the Sergio apparently fraudulent database. And there's also a commercial interest, I believe. This is my personal belief, right? There's an increase in demand that has, if you look at the Brazil data, for example, from last year, there's this flat gray line here, and there's been a 20-fold increase in demand just in one country. If you want to know more, I would recommend you go and look at the stock markets of some of the people producing related products and what happens after these open letters come out. So what's the science? There is good science coming up. There are 59 studies registered. I think Nathalie mentioned some 35 still active, and there's more coming up. There's good animal data from Pasteur Institute showing that if you treat hamster at the moment of infection with SARS-CoV-2, there is a marked reduction in olfactory deficit in these animals. And I think this elegantly proves some effect. This is seen at 400 micrograms kilo, but also with lower doses. There's also this elegant work from Argentina showing that there's a correlation between the Cmax achieved in patients and the rate of viral decay. Also small numbers, right? And then this is our own trial published last month. It's a pilot trial with very small numbers, but we do see reduction in the median viral load at 4, 7, and 14 days after treatment. Although, if you look at this, there's a huge overlap between ivermectin and control groups. But still, for hypothesis generation and to somehow shed some light whether additional data is needed, this was very interesting. We also see halving of the olfactory deficit, particularly in male patients, but in overall, there's a halving, so 50% of those of the olfactory deficit seen than in the control group. And there's this data from Bangladesh coming out with 400 patients showing clinical benefit. There will be data coming out of Israel with 100 patients also showing clinical benefit and also showing reduction in anosmia. But there's, again, negative evidence as well. Everybody was waiting eagerly for this N=400 study from Colombia, and it's not been published yet, but there's statements by the PIs showing or saying that there was no clinical benefit. There is now an evaluation ongoing by the WHO guideline development group, and hopefully something official will come out in the next few weeks. And what I see as the consequences for NTDs, I'll go in the last minute on what I see are key topics. And one is that for NTDs, this drug is used once a year at 200 micrograms kilo, perhaps 400 micrograms kilo, normally single doses. Those are advocating for COVID treatment may have to use it more than once a year at high doses, what I call high doses, and sometimes with multiple day regimens. For prophylaxis, and there's people advocating for this use, there's still to be defined whether it's pre or post exposure, whether you should use it more than once. It's also continuous use. You would have to use it periodically, right, or daily or weekly or continuous use. And there's still unclear whether we should go for higher or lower doses. Another point is the interface between COVID and NTDs. This is just to remind everyone that if you're going to give high-dose dexamethasone to someone in the tropics, please treat them with ivermectin regardless of its effect on COVID because of strongyloides. And we don't have good clarity yet whether deworming everyone will make COVID better or worse. But this surge in demand is also fueling worrying stuff like this study just published with a review of 7 products in South Africa, of which some of them contained undeclared substances, including benzodiazepines, pregabalin, or antiplatelet agents, right? And some of them selling for $70. There's also illegal sharing and counterfeit products in Latin America. People using veterinary formulations to inject thousands of people, including stuff that looks like ivermectin, but it's not quite ivermectin, like doramectin. And this is the take-home messages I presented before. I won't repeat them, and it's still yet to be seen. It's rolled out, evidence is emerging, but it's not quite definite to support broad programs yet.
Thank you very much, Carlos, for that. I'll just come back on. I thought that was wonderful. Thank you for that. I can see why they chose ivermectin for repurposing and not bleach, to put bleach through that entire process. And I actually wonder how much of the resistors that you talked about there, the contempt and these kind of things, these issues, are rooted in the rise of scientific populism, as we've just seen what happened in the last 4 years in the States. And I wonder if that's on the way out, and perhaps it's back to the real science. There's some very interesting numbers you're putting forward in terms of your own study and some of the impacts to the wider kind of dosing and resistance kind of areas for NTD. So it definitely needs more work, but I think maybe the environment now has changed in that direction. We've had quite a few questions come in, which I'm not going to ask now. I'll ask at the end. A lot of people making comments on such an excellent session so far and presentations. I'll just give a quick shout out. We've got Enoch Boateng from the Czech Republic's lab on molecular helminthology. Obviously, helminths are a primary area where repurposing is always going to happen. So he's coming in from there. We've got from the London School of Hygiene and Tropical Medicine, Mohammed Afolabi. Also from Nigeria, Emmanuel Agunloe. From the DRC Congo, Dr. Celestine Modugo. Nigeria, Yusuf Jovel Sani. From the Ministry of Health, Gabon, Dr. Anik Monjo, dialing in as well. We've got a very widespread from Argentina, Dolores Cara from CONICET, who will be speaking actually later in the leishmaniasis session. So a wide kind of spread of countries, which I think really does speak to the value and the perception of repurposing as a viable strategy. So it'll be very interesting to see how your presentation, Carlos, develops. Perhaps it's going to be part of the blueprint on how to actually do repurposing moving forward. So thank you very much for that. I think that brings us to the next speaker, and I'm very happy and honored to give the floor over to Dr. Luciane Tritton from the University of Zurich, where the paper really was about when secretomes meet anti-helminthics, lessons for therapeutic intervention. The reason we had Dr. Tritton here for this particular session, we've heard before the kind of larger impacts of repurposing from a very strategic perspective from Nathalie at the DNDi. We've heard Heather's more kind of operational, functional role of Cure ID in bringing groups together and getting that information flowing all the way to RCT. And then we've heard Dr. Chaccour giving us a case study really on ivermectin in the age of COVID, some of the barriers to that and some of the potential positives that came out of that process, and it's still ongoing. We thought we'd bring Dr. Tritton in simply because we had an ISNTD Connect session, I think about a week ago, which looked at onchocerciasis-associated epilepsy. And part of that was that the method, rather the mechanism of action for this onco-associated epilepsy with measured prevalences, peer-reviewed data, all of it was very, very misunderstood. And for those of you, obviously, we all know that it's the decrease in 5-hydroxytryptamine and the decrease in the 5-HT levels, which then themselves will go on to trigger anxieties and trigger epilepsy, or the spectrum of events on the epileptic spectrum. And the secretome in that setting of onchocerciasis from the parasite itself could have been a modulator that was modulating downstream the 5-HT levels in the brain. And so this whole area of the secretome does present itself quite well to repurposing in terms of new targets, new areas to investigate. And so we thought we'd bring Dr. Tritton in for that. A long introduction, Luciane, you have to forgive me. I'm going to hit on that basis. I think round of applause.
All right. Thank you very much for the kind introduction. So very pleased to be here today. And indeed, I'm going to take you to a slightly different kind of story. As we all know, the expansion of the armamentarium to control helminth infection is urgently needed. And so is the acquisition of a better, deeper understanding of the mode of actions of the currently used anthelmintics. So with this, I will take you through the very recently published opinion paper that was called exactly as this presentation, When Secretomes Meet Anthelmintics, Lessons from for Therapeutic Interventions. So to survive in their hosts, humans have evolved a range of strategies to remain there for months or even years. And you can imagine the host environment is not a very hospitable and friendly one when there is an engaged immune response against the pathogen. So there has been a molecular negotiations between host and parasites. And this has resulted in highly specialized interspecific relationships in which the secretomes may delimit conditions that render a host permissive for an infection to take place. So what are these secretomes? These are basically any kinds of molecules that are released either by excretion or secretion by a parasite into the host environment. And these may be proteins, nucleic acids, lipids, small metabolites, glycans, and basically anything. And this also includes extracellular vesicles, which is a very fancy research topic in molecular helminthology nowadays. These are small membranous structures that are released by any kind of cell. Virtually any eukaryotic cell releases extracellular vesicles, and these are like small compartments encapsulating all these other kinds of proteins, nucleic acids, and other molecules. And so many of these secreted molecules or excreted molecules have the capacity to modulate host immune responses. And these have attracted, obviously, increasing attention. So these secreted proteins have consequences, functional consequences for host cell functions. And these consequences are very varied. It really depends on the type of cell we talk about and also the type of pathogen we talk about. But obviously, secretomes are a promising source of drug targets, both for drugs and vaccine discovery. So obviously, identifying parasite-derived molecules that render a host permissive for an infection to take place should encourage efforts to rationally target these important key components to restore the host's capacity to either resist or expel, eliminate the infecting worm. So an anthelmintic drug discovery in real life is done like this. So initially it was done in the early years, in the 70s and so on, by simple, if I may say so, empiric observation of efficacies in vivo, so in an infected animal. And this has shifted slightly now to more modern days to phenotypic screens, mostly ex vivo, in vitro, where the viability of a worm is observed as a consequence of exposure to a drug or a compound. This has been complemented partly, at least, by mechanism-based screens. So this involves the availability of recombinant systems and powerful in vitro tools. This is, however, slightly limited by the fact that the mode of action of many drugs and compounds are unknown. But screening a little bit the literature, we noted that there was an underappreciated characteristic of some anthelmintics, that is their ability to block the release of these parasite-derived molecules, so that these anthelmintics are basically capable of disrupting this dialogue from the parasite to the host. Do anthelmintics act by disrupting parasite secretory or excretory processes? Well, basically, the characterization of parasite-derived molecules has led to the concept that therapies can be devised to disrupt the dialogue between parasite and host, and work on vaccines that specifically target secreted antigens based on their known or just presumed roles in enabling an infection has shown that this approach is valid and can generate therapeutically useful outcomes. Now, there is evidence out there in the literature regarding the macrocyclic lactones, so I'm going to talk about ivermectin again. And this class of drugs acts by interfering with Helminth's nervous system by preventing responses to excitatory neurotransmission. And this generally results in an impaired muscle function and paralysis of the worm. Where the benzimidazoles, where albendazole and mebendazole are classified, act rather by inhibiting the microtubule polymerization. And this results in deep physiological alterations of really multiple, basically any processes, and that includes secretion. So the absence of stable microtubules hinders the intracellular trafficking of these parasite-derived molecules and their ultimate secretion. If the mode of action of some anthelmintics seems to impair aspects of parasitic nematodes that control these molecule releases, then the effects on secretion seem to be correlated very well with the spectrum of activity and the kinetics of expulsion of infecting worms, as well as with the levels of drug exposure that is achieved in vivo. At least this was shown for a few nematode species and a few stages. But whether inhibiting secretion is the key trigger for parasite clearance or one of several pharmacological effects remains to be determined. The benzimidazoles have a only not very obvious effect on adult worms of several nematode species. So where the parasite motility, their viability remains largely unaffected. In fact, it is a disruption of parasite secretory processes that seems to be the earliest physiological effect of this drug class exposure. This is translated or illustrated by the accumulation of secretory granules along with disappearance of microtubular structures. That's what observed at least in Ascarisum. And there is also an accumulation of certain proteins, such as acetylcholinesterase in parasites. And this was noted along with a marked suppression of the release in vitro of this very protein. So these effects observed with benzimidazoles at least applies very well to gastrointestinal nematodes. The situation is a little bit different for tissue-dwelling nematodes because the expulsion kinetics are partly the result of the duration of exposure to effective drug concentrations. Plus, the action of benzimidazoles is reversible. So the pattern of exposure to benzimidazoles and the dynamics of parasite expulsion are compatible with the contribution of disrupted secretory processes as a cause of parasite elimination. Now, for the macrocyclic lactones, these mainly kill or affect developing stages, so microfilaria in filarial nematodes or embryos. And there is evidence that has accumulated to support that the role of the ivermectin targets, the glutamate-gated chloride channels, in parasite-derived molecule release via the control of neuromuscular activity of the secretory system. And it was shown that in Burgdorferi microfilaria, these ion channels are associated with a structure surrounding the secretory vesicles. And this is proposed to control molecule release. Now, both protein and extracellular vesicles release is inhibited by macrocyclic lactones. There is now even more evidence accumulating regarding the effect on these class of drugs and the production and release and the contents of these extracellular vesicles. Now, what we suggested is that the identification of key parasite-derived molecules as determinant of the equilibrium between host and parasite, as well as the mechanisms that control their release, will likely expand our list of targets that can be exploited for therapeutic interventions. Now, the evidence from pharmacology and immunoparasitology as well should be reconsidered and put together to inspire the search for novel ways to interfere in the communication between parasite and host. And we would like to recommend that efforts be made to incorporate screens for inhibition of these parasite-derived molecule release in culture as a complement to existing phenotypic screens. And there are very famous technical challenges to do this that may limit our ability to pursue this goal. And this is, for example, our incapacity to maintain the complete life cycles of these nematodes in vitro. We still heavily rely on animal models. And our current in vitro systems are flawed, but these are the best we have. And we'll also recommend that expanding the scope of discovery programs, so that these integration of product release and phenotypic screens may help really greatly expanding the scope of discovery program to be able to detect such compounds. And that is going to be an investment, but that should be an efficient one to bring a new compound into the pipeline. I'd like to finish with a couple of outstanding questions, of course, that should or hopefully will shape research priorities regarding new anthelmintic discovery. Can we ultimately reduce helminth secretory systems to an in vitro model to complement currently used phenotypic screens, so in vitro systems? Will it be possible at all to reconstitute the host parasite crosstalk using in vitro systems and maybe applying a stress situation that mimics what the host does to the parasite to reach a setting as natural as possible, and this to be integrated in anthelmintic screens? And can we develop tools for functional genomics of nematodes that will facilitate the characterization of parasite-derived molecule secretion biology as well, and then identify, prioritize molecules that are essential for establishing and maintaining an infection? Now you can find basically all I said in the paper entitled When Secretomes Meet Anthelmintics, Lessons for Therapeutic Interventions, which is published in Trends in Parasitology. And I would like to thank very much my co-authors, Dr. Giovanni Moreno and Professor Tim Geary, as well as our funders, and I'm very grateful for your attention.
Thank you very, very much, Lucienne. That's wonderful. Real new horizons opening up there in terms of the direction of travel for people involved in repurposing, elucidating the mechanisms involved, but using this secretome approach. Some very strong recommendations you put forward there as well in terms of the reconsidering pharmacology, the pharmacological evidence base, and the host immune evidence base that's there, and as well as introducing these PDM screens at the phenotypic screening level, reshaping the way we assume things. I'm sure there'll be a lot of crosstalk between yourselves and some of the other people. I know there's the helminth elimination platform with the oxfendazole repurposing from neurocysticercosis settings into beyond. We'll have a lot to talk about, I'm sure, in the Q&A, but thank you very, very much for that. Wonderful.
Thank you.
Brilliant. I think you really complemented and it steps with everybody else's presentation as well. That brings us very neatly into a segue for the last speaker or speakers. Very honored to call Dr. Alvaro Acosta Serrano a friend of the ISNTD from the Liverpool School of Tropical Medicine. There'll be two speakers, Dr. Alvaro Acosta Serrano and also Dr. Laith Jacob from the London School of Hygiene and Tropical Medicine. So it's Liverpool and London together. Watch out. It's going to be good. I'm going to hand you over to Alvaro.
Thanks very much, Karen. And it's always a pleasure to talk to ISNTD and support the activities that you and Marianne do all the time. I think you guys do a great job. So thanks for this invitation. And this is going to be very different to previous talks I've given to this audience on leishmaniasis. This is on our program on trypanosomiasis and tsetse flies and trypanosomes in general. And this is all about something that is being sort of discussed in this session, but not usually we think about drugs that will control bacterial transmission of any NTDs, in particular this one, trypanosomiasis. So I just want to share this story, and at the end, my colleague, Dr. Laith Jacob, will continue with this. So I think this audience doesn't need to much of this introduction, but just to put things into context, we are in a great time where human African trypanosomiasis is about to be eliminated. So this is a projected graph from the WHO, perhaps a little bit old, but that was the projected number of cases, and we're actually very close to it. Most of the cases are concentrated in few countries, in Democratic Republic of Congo and Uganda as well. And so this is on the verge to be eliminated fairly soon, we hope. And that's the result of a combination of better drugs, better and safer drugs, better diagnostics, to control trends. So what will continue to be a problem beyond having to control human Hamilton trypanosomiasis will be the animal trypanosomiasis situation, because this is, we still don't have solutions, we don't have vaccines, there is a lot of drug resistance involved in that, and there is a great deal of animal trips that are transmitted by other insects, not necessarily tsetse flies. So we need to come out with better and safer vector control tools for this. So what I'm going to talk about is in relation to something that is how we can exploit the physiology of the fly. And in this video made by a Korean group, taken from our facility at the Liverpool School, you can see a tsetse fly landing on the producing arm to take a blood meal. And although this is fast forwarded, this process takes about 10 minutes, and within those 10 minutes, the fly takes up to 30 macroliters of blood. So within that process, approximately, the fly expands twice its own volume. So this is quite remarkable. This is to think a human weighing 50 kilos will have in one go 100 liters of milk. So blood-feeding insects do this, and do this for several purposes, but one of these to avoid visitation, so they don't need to take a blood meal all the time. But also they have developed this, and in order to they have developed fast digestion system in order to really degrade all these products, molecules contained in the blood meal. So a few years ago, this man, Marcos Terkel, who at the time was a postdoc in the lab of Pedro Oliveira in Rio de Janeiro, he was studying these particular aspects of blood feeding, and what he found is that for several arthropod vectors, he found that one molecule, one metabolite, tyrosine, that comes in the blood meal is essential for the life of those insects for different reasons. But he also found that when he does, when he blocks that pathway, either genetically or pharmacologically, then the bugs die, or they have severely compromised the reproduction system. So Marcos came to my group on several occasions and did a fantastic job. And the idea was to whether the same approach to inhibit the tyrosine degradation pathway will work with Glucina, and whether that then, as an academic concept, could be exploited in a more translatable way to control tsetse population in endemic areas. We're not that far at the moment, but we think this approach will have a strong potential. So this particular work just been published in PLOS Biology a few weeks ago, and involved not only Marcos's work, but also a great job by Lee Haynes, a postdoc in the lab, and also several collaborators, many to mention right now, but Leif at the end will mention, but Leif Jacob did a great job into all these mathematical modeling that I will explain in a few minutes. So I will just go through several validation experiments that when we start with something from scratch, and we need to know, and just to make sure that will have a potential to be applied in the field. And one of those, first question is whether if we do genetic ablation of the genes involved in the tyrosine degradation pathway, would that have an impact in tsetse? So in order to do this, we did RNAi knockdown. So we injected double stranded RNA into the thorax of tsetse fly, and we used 2 genes encoding for 2 important enzymes in that pathway, TAT and HPPD in short. And when we did that, then compared to the control, which was just PBS injection, we see that there is a huge decline in days. Oops, I did something wrong here. Okay, so days post-injection, you can see that there is a decline in the survival of those flies. So that's a genetic validation that that pathway is essential for the fly when they take a blood meal and have those genes knocked down. Then we went to the pharmacological aspects of it. So we tried several inhibitors of the tyrosine degradation pathway. One of those is a very well-known herbicide produced by Syngenta. It's called mesotrione. Mesotrione is a weed killer. Syngenta sells millions a year using this product. And what it does is to inhibit HPPD. Mesotrione has an effect, and so I'm not going to show this result, but the one that is actually at least 200 times more potent than mesotrione is this one, is nitisinone. So nitisinone is another one of these family of herbicides that in the 80s and early 90s was repurposed to treat one genetic disease that had no solution, that is tyrosinemia type 1. And this is a disease that also affects the tyrosine metabolism pathway in babies. If these individuals don't take nitisinone for the rest of their life, they die, basically. So this is FDA approved, and it's being used since then. There is another recent one, another disease called alkaptonuria. Alkaptonuria is another metabolic disease that has defects in the tyrosine metabolism pathway. And a group at the University of Liverpool, led by Professor Ranga, they have worked out that nitisinone can also be used to treat this disease, and this has just been approved as for last year. So in order to then how we can assess this in Glucina, so we had we're very fortunate to have a large colony in our institution, so we just spiked the blood meal with several concentrations of the drug. And what we found is that in a concentration-dependent manner, we see a strong inhibition of the survival, so basically death, and even using up to 20 times lower concentration that are used in individuals that are affected by either tyrosinemia or alkaptonuria. So in order to continue with this, then obviously the killer experiment was to treat animals. So we treated animals with the drug and then exposed them to the virus tsetse fly. We used 2 different concentrations that are matching what are administered to humans for these diseases, and we saw actually decline in a very similar manner to what we see in artificial blood feedings. Another interesting aspect of this is that the drug could be used topically as an insecticide, so it's highly soluble in organic solvents. So, for example, acetone. And when we do that, for example, when we apply the drug in to the fly, they don't die immediately, but the minute up to 144 hours at least, they receive a blood meal, they immediately die. So the death mechanism is active once they get exposed to the blood meal. So there are many things that obviously could be happening. We want to understand this to understand potentially, well, the mechanism itself, but also potential resistant development. So using metabolomics with colleagues at the Craig Institute, we validated that this drug actually increases the level of tyrosine. So this is a time course of the experiment. So tyrosine gets accumulated. Another metabolite, hydroxyphenol lactic acid, which is in human involved in acidosis, metabolic acidosis, is also highly increased. And then as a control, if we look at fumatic acid, which is one of the end products of this pathway, then after 10 hours, it gets completely shut down. So that is biochemistry validation that we are hitting the right pathway. So what natisinone does to the fly, it does many horrible things. It starts a few hours with paralysis, so it affects flying muscles, but then it leads to a systematic tissue destruction by inducing a systemic melanization of the insect. In the meantime, there is a changes in eye coloration, so the eye becomes golden. This is the changes in coloration in the abdomen, as I said before, and also samples from different places of the treated fly, fat body, and fly muscle. You can see the melanization. This is only a few hours before they die. So one important aspect to this is that, as I said, this gets only activated by when the insect gets in contact with blood. Okay, otherwise that doesn't happen. And importantly, we wanted to make sure that pollinators, like bumblebees, they get affected. They don't get affected by the drug. And with these experiments in collaboration with the group at the University of Liverpool, and what we found is that when we expose bumblebees to different concentration quite high, and those that induce a killing phenotype directly in tsetse, we see that compared to the PBS control, there is no effect until several days. So at least they don't induce death in pollinators, which is quite important. And this is unlike some of the neurotoxic insecticides that are although we still don't know whether they will affect in the reproductive system. This is a pending experiment to do. So beyond tsetse control and for other entities, so would this concept will work for other blood-feeder insects? And the answer is yes. So we have tested this for Anopheles, and it worked very well. And in fact, we're looking into those aspects. Our collaborators in Brazil, they had a paper, a preprint paper that showing that it works for Aedes aegypti, but also for local Anopheles species affecting Brazil. We have tested and not published yet the effects on phlebotomins. Again, our collaborators in Brazil have shown that ticks are also highly susceptible to the drug. So that means that, for example, in terms of veterinary use, if we treat animals in farms in Africa, for example, to control trypanosomiasis, that could also affect, for example, the local populations of mosquitoes and ticks, because obviously they're constantly feeding on cattle as well. So just to summarize this before Leif start with his talk, we know that I hope I'll be able to convince you that natisinone can be used to kill tsetse flies by inhibiting the tyrosine pathway. This drug can be used, it's ingested in a blood meal from host or topically or through the cuticle. So the cuticle aspect is very interesting as well and exploitable for many reasons. The fact that they can be ingested along the blood meal further develops this concept of what is an endectocide. And at the beginning, I wasn't really convinced to go on that route. But then a recent collaboration with Gaith Al-Yayusi from the Liverpool School and with a PhD student from my group, Anna Tret, they have looked into the PK/PD profiles of natisinone in comparison, for example, with ivermectin, which is the gold standard. We can see that natisinone has a superior PK/PD profile. Does that mean that it's a better drug? I don't think so. It's a long way to go. and we still have to sort out some problems potentially with toxicity. This toxicity comes from the fact that patients with tyrosinemia or camptonuria, they have to take the drug on a daily basis, and so they have some of the consequence. But in terms of vector control approaches, we're looking into a single dose that will be compliant with the MMV guidelines, and will be used in combination with antiparasitic drugs as well. So this is currently in preparation. So this approach, as I said, it could be used as an integrated vector control package because it will also affect other blood-feeding insects. And importantly, it's ecologically friendly because at least we demonstrated it doesn't kill pollinators. So an important aspect of this work as well is whether that might work in the field. In the case of trypanosomiasis, for example, African trypanosomiasis, we got that 2 different parasite species that are transmitted by 2 different glossina species and in different ecological situations are responsible for sleeping sickness, although the Gambiense one obviously is the most important one. So that's when the important work of my colleague Jacob into looking into this mathematical model in the field, and also to validate whether this approach can be used. So I will leave now, I will stop sharing and leave this with Leigh's presentation.
Hello, I'm Leith Jacob and I work at the London School of Hygiene and Tropical Medicine. And I'm here to talk about some of the modelling work we did to complement the lab work produced by Alvaro and colleagues on nitisinine. And I just wanted to share this picture with you. This is a sculpture outside Keppel Street building of the London School of Hygiene and Tropical Medicine. So the next time you come to the London School, please do keep an eye out for it. It's Tsetse fly. So the way that we conducted the modeling was by using a Ross-McDonald equation. Many people will be familiar with Ross-McDonald for vector-borne disease modeling. It's been used predominantly for understanding malaria transmission, but it's equally applicable to most vector-borne diseases. And at its simplest, it is R0, which is the basic reproduction number, as derived from 5 key parameters, and they are M, which is the number of flies per person, A, which is the daily bite rate, and it's squared because there are 2 bites required for the pathogen to complete its life cycle. P to the N is the probability of surviving N days, R is the recovery rate, and mu is the fly mortality rate. So for example, if you wanted to simulate larval control, the way that you would do that would be by reducing M. So you reduce the number of flies per person, and that reduces R0. If you wanted to simulate treating the sick, the way to do that is by increasing the recovery rate. So you increase little r, and it's in the denominator, and so that reduces the overall R0. Now, for almost 100 years, people have appreciated that killing adult vectors is normally the way to go, and the way we understand that is because when you kill adult vectors, you have a multiplicative impact on multiple parameters. So in this circumstance, we're not just reducing the flies per person, M, but we're also reducing the probability of surviving N days, which is Pn. And we are increasing the fly mortality rate again in the denominator. So you have this multiplicative impact on transmission. Taking the efficacy data from Alvaro's work and also the longevity of its efficacy, the half-life of nitisinine and its efficacy, for example, we plug that into a simple Ross-McDonald model to inform its impact. And this plot is just showing the RE this time. RE is the effective reproduction number, which is just the basic reproduction number, but under interventions. On the y-axis, we have RE, and on the x-axis, we have time in days. And this is just a simple simulation of monthly dosing. So with monthly doses, we get a drastic decrease in the reproduction number, followed by a gradual increase where it re-equilibrates at its equilibrial level. pre-control equilibrial level. And you get these characteristic monthly pulses of RE. And the different lines are just simply denoting different efficacy levels, or different coverage levels, actually, different coverage levels. So one of the nuances of tsetse flies is that they are really inconsistent with where they source their blood meals from. So depending on where the study was conducted, or how it was conducted, or when it was conducted, the majority of blood meals sourced by tsetse flies can have rat origins, or it can come from bats sometimes, or other wildlife species. Some tsetse fly biting behavior studies have identified humans and livestock as the sources of most blood meals. And the reason why I thought it was important to incorporate this kind of nuance is that whilst it's feasible to dose livestock and even human blood meals, or human blood, with nitisinine, NTBC, it has FDA approval for use in humans. It's less feasible probably to get that drug into the blood of wildlife. And so we needed to account for that. And the way that we did that was we simulated all the possible combinations of blood meals there is, split between livestock, humans, and wildlife, under the assumption that we would never be able to get the drug into wildlife blood. So here we have some simulation results looking at dosing livestock. Only on the y-axis, we have the proportion of bites on livestock. So at the bottom, no bites on livestock, and at the top, all bites of tsetse flies go onto livestock. On the x-axis, we have the proportion of bites on humans. Again, 0 means no biting on humans, and 1 means all bites are on humans. And so, for example, where we have 0.2 bites on humans, 20% of bites on humans, and 20% of bites on livestock, that necessarily means that the other 60% of bites is happening on wildlife, and we can do nothing about that. Now, we have 3 plots here, and they are demonstrating what happens when you treat livestock every 10 days, in the middle every 30 days, and on the right every 90 days. And the color is corresponding with the effective reproduction number. So the brown regions is where we are failing to break transmission, where the effective reproduction number is remaining above 1. And you can see when we have very frequent dosing, so in the order of every 10 days, and provided that at least 20% of the blood meals have livestock origins, then we are actually able to break transmission just using this as a standalone control strategy. But again, at very high frequency. So the equivalent plots for dosing humans, it's better for breaking transmission. And in this circumstance, when at least 20% of the blood meals have got human origin, you only need to dose humans every 30 days in order for you to break transmission. And then, as you might expect, if you can dose both livestock and humans, then provided at least 5 to 10% of your blood meals have got human or livestock origins, then you can enter this white or blue region here, and you can break transmission. So that's an encouraging result. So very quickly to conclude, we feel that host targeting needs to be informed by local tsetse biting behavior. If local tsetse are sourcing the majority of their bites from wildlife that you can't drug, then this is not a viable control option, probably. We need to emphasize the point that the current half-life of nitisinine is quite short, and that massively constrains the sustainability of this. We don't envisage NTBC treatment to be a standalone control tool to eliminate transmission. We envisage it to form part of an integrated control to achieve elimination. So in the final stages of achieving elimination, if you're struggling to break transmission, then NTBC could be a good complementary tool to use. And I think it was the general conclusion of the most recent WHO meeting of stakeholders on the elimination of GAM-BNC that an integrated approach using novel vector control tools such as this is the best hope that we have of eliminating HAT. And with that, I just want to acknowledge all our fellow collaborators and co-authors on this huge endeavor. And thank you very much for listening.